Okay, guys, let's do it. Okay, lecture 3. Here we are. Who thinks that this class is, this lecture is unnecessary? Raise your hand. I just found out someone thought my lecture was unnecessary. That's the funny thing is when you say things to people, get back to people. Anywho, so quick business. Don't forget to take the pre-test. It's due tomorrow. Okay. I like to know how you're doing, and it's 10 points. So don't forget to bring your Scantron presentation today. Don't forget, there's a quiz this weekend. A lot of people actually forgot last weekend's quiz. You guys should be going, oh, right now, and put a reminder in your phone. If you had a reminder in your phone, you wouldn't forget. I swear I forget everything. That's what keeps me going. I would forget to, I don't know, we won't even go into the embarrassing things I've forgotten. Okay, quick, Stump the Teacher. So the question was, what happens to the signal sequence in the ER after it is cleaved? I got a lot of great answers. Turns out the answer is in the textbook. So that makes it really easy to find. So that's always a good place. You guys are great for looking there first. But basically, what the textbook says is pretty much it comes out of the core and is degraded, which is kind of what we thought anyways. But then there were some papers out there that talk about how actually there's some function to the signal sequence, and it can act in a signaling sort of way as some sort of receptory kind of thing. So that was cool because I didn't know that. So thank you all. Those who got it right got a point. I was working on how to do it through Blackboard, and I think I can set it up as an assignment, but that would require me to move really quickly after class. So we'll see if we can get that together. Questions on that? What it meant, what we're talking about, where we are? Okay. Okay, so we're going to take 5 minutes to write. This is our first question that's going to kind of look like an exam question, so hopefully this will be really helpful for people. Okay, TNF-alpha is a cytokine secreted by cells in response to infection. It plays a role in activating the inflammatory response. The gene encoding TNF-alpha is transcribed, processed, and exported from the nucleus. What happens next? Hint. Include all steps relevant to Tuesday's lecture. So don't tell me what you learned in 201 and 309. Tell me what we've learned in this class that's relevant to this. Okay, so take 5 minutes, do the best you can, and then we will go over it. Yes?
What was the copy on the questions when we're writing?
I don't care. Whatever you want to do. Really, the purpose is more for you than anything. In the end, I will make sure every page is dated. But I have this, and then you'll have it on the lectures online. Yeah, don't copy it, actually, because that'll take time, right? Don't do that. Okay. I see a lot of people looking in their notes. You should do that. That's helpful. Okay, I think that's about 5 minutes. So for those who've been walking in late, you guys are missing out on this, which is unfortunate. And I'm seeing you guys. So if I keep seeing you guys, then at the end of the semester, I will notice that the 5 points that you're getting for this might not be for you. But anyways, okay. So I'll take some people's answers, but first I want to kind of teach you how to take this question apart and figure out what it's really asking. Your first instinct might be, I don't know what TNF-alpha is. I'm not an immunologist, right? Well, don't worry about that. Those are all distractors, okay? What you need to do is pull that information out. TNF-alpha is a cytokine, blah, 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 blah. Secreted. That's your key word there, right? It's secreted by cells. So right away, that should trigger secretory pathway, right? It plays a role in whatever. The gene is transcribed, processed, exported from nucleus. What happens next? So who wants to share with me what they wrote in a way that doesn't take too long? Who's brave? I can call names. It's okay. Unless we have a Melvin here. Yes, Mackenzie.
So a ribosome will attach to the mRNA and start translating it, and the signal sequence will be translated, and the SRP will attach to it, pause translation, take it over to the ER, insert it into a 4, leave so that translation can start again, and then the protein gets directly translated into the ER, and the signal sequence gets cut off at the end.
Great. That's actually a perfect answer. So I'll repeat it for those on this side of the room who may not have heard. Right. So the mRNA is going to associate with the ribosome. Then translation will start. Signal sequence will be translated. Translation will pause because the SRP is going to bind to the signal sequence. Pause translation. Bring it over to SRP receptor. Then it will go into the translocator in the ER. That's the one word that you have missing, but it's a port. Yes, you're correct. You would lose points for that, but it would be super perfect if you said translocator. And then translation will, then SRP will go away, translation will continue, and then the signal peptidase inside the ER will cleave the signal sequence, and you'll have your protein in the ER. And that's as far as we've gotten. There's more things that are going to happen after that, but this was just the steps that we've learned about. How many people knew exactly to go to that general theme? Raise your hand. Okay, good. That's good. How many people had no idea what to do because they were distracted by TNF and cytokine and all that? Good. Don't let yourself be distracted. Okay. So hopefully by the time we get to the exam, which I just checked is like the 30th, keep that in your mind that it's still like 4 weeks away, but you know, it's going to come eventually. Hopefully by the time we get to there, we'll have gone through so many of these that you won't be shocked from the questions that are on the exam. Okay, let's learn something. Yes?
When does co-translation happen or post-translation?
Okay, so I got the question about when is co-translational versus post-translational happening. Depends on the protein. As a rule, since we spent so much time talking about co-translational import, you would be smarter to focus on that and answering a question. Always think to yourself, what did she talk about for 1 second, and what did she talk about for a whole lecture? And that's what you will want to talk about, but it would be even a super duper answer would say sometimes you also have post-translational import in addition to what we just talked about. Good. Any other questions before we move on? Okay, so far. So I always have this so far slide, which kind of summarizes what we've done already, which you kind of just summarize yourselves in your brief writing. But as the unit goes on, it gets longer and longer, and you can kind of think of this as the things that you should be focusing on in your studying. So far we started our unit on intracellular traffic. And that's just generally the trafficking of proteins to the correct location in the cell. And we started with the secretory pathway, and this is for proteins that are destined for secretion, plasma membrane, and lysosome. So I could have easily put any other kind of protein in that question. I could have said an acid hydrolase, which lives in the lysosome. I could have said the LDL receptor. Any of those things could have been interchangeable distractors. Okay, so you should know, of course, the role and function of the signal sequence, SRP, SRP receptor, translocator, signal peptidase. So that kind of tie it all together. If you find yourself just memorizing what each thing is and not tying it together in a process, come see me, because a process is more like a story that you can remember rather than random details. Then we spoke about how integral membrane proteins end up in the plasma membrane. We spoke about the stop transfer sequence, or an internal signal sequence that acts as the stop transfer sequence, which dictates where that transmembrane domain is going to be. And so that's where we're going to start again today. So we spoke about the stop transfer sequence, and that shows up when you have a signal sequence, and then you'll be targeted to the ER, you'll start translating again, until you hit that stop transfer sequence and then move into the membrane. This is a different situation where you can have an internal signal sequence, which is kind of acting like a stop transfer sequence, and that's going to be your transmembrane domain. And it could be integrated into the ER membrane in 2 different ways, basically just with 2 different topologies, right? In one case, your amino terminus is on the cytosolic side. and in the other case, your carboxy terminus is on the cytosolic side, which matter. It really, in the sense of what you guys are learning here, it doesn't really matter. You just need to know it's one or the other. But if you were a researcher studying a specific protein, it would matter because you would want the signaling portion to be on the cytosolic side, and you would want the extracellular portion of the receptor, because it's a poly receptor, to be on the luminal side. Okay, because that's where they're going to need to function once you get out of to the plasma membrane. Okay, and so I'm going to keep mentioning the word topology, and that's really what I'm referring to is what's on what side. And remember, it's going to stay all the way to the plasma membrane. So this is just kind of a zoom in of how that looks, right? So you would have your internal signal sequence, right? That's acting as the stop transfer sequence, and that's going to bind right here. You're going to have continued translation, so that's going to kind of drop down, and so your carboxy terminus is on the luminal side. And then here, the same thing's going to happen, but the only difference is that this your amino terminus is going to end up on the luminal side. Right, so topology is maintained through the whole secretory pathway. Why is it important? I'm only going to harp on it a little bit. So if you think about the secretory pathway, right, you're starting in your ER, then you're going to your Golgi, then you're going to your plasma membrane, right? And so if you look at any, let's see, I'll use, is that going there? If you pick any protein to follow, so let's pick this squiggly pink one here. So you'll notice on the luminal side, it's a lot longer than on the cytosolic side. Actually, should we use that? Yes, let's do that one. And so you'll notice what's going to happen is it's going to bud out of here into a vesicle. And when it buds, this squiggly side is going to stay in the luminal side. And so you can kind of see here. Here's your vesicle. And we're going to talk all about budding and vesicles and targeting and all that stuff. But notice it's staying on the luminal side, and it's going to fuse with the Golgi, and it's going to go through the Golgi and do what it has to do in the Golgi, all the modifications of sugars and whatnot. And then you are going to make your way to the plasma membrane. It's not really showing, well, whatever. So notice here, so what would happen is you could imagine this vesicle up here that is going to the plasma membrane. Now notice it's containing something that's in the lumen, it's not in the membrane, so obviously that protein is going to be secreted. And you can kind of see right here, it's fusing and being secreted. But let's say it contained this receptor that we've been following, right? It was in the luminal site, so it would have been in the center of the vesicle, and as it fuses, it ends up on the exterior of the cell. And so you can take some time when you're studying to draw this out, and you can see how each fusion, your protein's not being sucked out of the membrane and putting back in, right? It's staying in the membrane the whole way through. So it needs to go in the right way in the ER, and it's going to stay that way all the way to the plasma membrane because you really want that ligand binding part of your receptor outside of the cell. And you really want that signaling part of your receptor inside the cell. Okay? Does that make sense to people? People always get confused on this, so I'm spending a little extra time this year in this concept. And so this is kind of another way, this was another picture I put in. Again, you can see here your ER is over here, and you have on the cytoplasmic side, that's your bigger, bulkier side. And notice, as it buds, it's going to stay on the cytoplasmic side all the way through the Golgi, and then all the way to the plasma membrane. Still, that must be the signaling end of your receptor. Okay? Are we good? Are we happy? Was people staring at me? People are always staring at me. What if I got paranoid? I was like, we're all staring. Okay. That's kind of my job, is to be stared at. Okay. So you can use radioactivity or fluorescence or anything to follow some sort of label to follow proteins through the secretory pathway. So you can imagine, let's say you put a protein of interest in. For the sake of safety, we won't use radioactivity here. We'll just talk about, here's radio labeled, let's talk about GFP, right? Green fluorescent protein, that's something you can think about very easily. And you can put your GFP in and just make sure it has some kind of transmembrane, or not transmembrane, some signal sequence attached to it, so it goes to the ER. And you can follow it over time. 3 minutes, it might be in the ER. By 7 minutes, it might be at the Golgi. And by 120 minutes, it might be secreted from the cell. So those same proteins are making their way through that secretory pathway. If it was radio labeled, you would be detecting by probably like a Western blot. If it was GFP, you would be detecting by immunofluorescence. So you can see that GFP. A Western blot, you can actually isolate your ER and your Golgi and then look in the supername to see if your protein made it out of the cell. Any questions about this? Yes?
Did you say that in order for like a protein to go to the ER, it has to have a signal sequence?
Yes. And that is the take-home message of these few lectures. So let's take that question one step further. So the question I just had is, wait, so in order to go to the ER, you have to have a signal sequence? Absolutely. Let's say you want to do this experiment with GFP. That's why I said you have to add a signal sequence, because GFP, as far as I know, does not have a signal sequence, but you can engineer it to have a signal sequence just while cloning by putting the sequence in your primers. And then you can send anything to the ER just by putting in a signal sequence.
Is the signal sequence always going to be the same, or are there different codes for it?
Is the signal sequence conserved? That's your question, right? Or are there different sequences are there different signal sequences? I think they're fairly conserved because if you think about it, how many SRPs are there? I believe there's probably only one SRP. Maybe there's more. Maybe there's a few. But let's say for the sake of diversity, let's say there's only 2 or 3. I can't see think there'd be much more. They have to all bind to signal sequence, and signal sequences will be on every protein that goes to the ER, so it has to be conserved so that same machinery can be used. Actually, in the paper that we did in recitation, they looked at SRP, they looked at the RNA component from different species, and it was fairly conserved even among species. So one would guess the signal sequence would be conserved. Okay, good question. Any other questions? Okay, so let's just go a little bit further with membrane proteins. So this is kind of a big picture view of how an internal signal sequence would be used to put a protein into the membrane. So here, you're going to have your mRNA and your ribosome translating, and that red part is your internal signal sequence. So actually, remember, translation starts at your amino terminus. So this much translation is happening before you get to your internal signal sequence. And then SRP is going to bind it and bring it to SRP receptor. And then it will interact with the translocon. And then you're going to get continued translation until the end. And then the amino terminus, in this case, will be on the cytosolic side. And the carboxy terminus will be on the luminal side. Notice there's no cleavage of the signal sequence because it's acting as a transmembrane domain as well. Okay, so remember we looked at this hydropathy plot the other day. Well, it was a different one. But what do you think this protein might look like in the plasma membrane? So you can think to yourself, well, there's a signal sequence here, right? And then a stop transfer sequence here. And notice it's very hydrophobic because it's pretty high above the 0, the midline. And it's probably about 10, 20, like about 30 amino acids long. So one would guess that's probably a transmembrane domain. We know that because it's been highlighted stop transfer sequence, but let's say it wasn't, you can kind of pull that from just how it looks. You could think to yourself, well, this is a signal sequence that's probably going to be cleaved because it's N-terminal. So it's not like that example of a signal sequence that's acting as a stop transfer sequence, because that would be more internal. If this wasn't here and only this was there, you can say, oh, that's probably a signal sequence that's also a stop transfer sequence. So my guess is probably this would bind to SRP and be brought over and interact with that translocator. And then you would get further translation until you hit your stop transfer sequence, and then it would move into the plasma membrane. And so if I put this on as an F side, that would be kind of the way you would approach it. And so it would look like this, right? Because here's your N-terminal signal sequence, and it's going to interact here with your pore, and then you're going to get continued translation until that stop transfer sequence is going to interact with the pore, and then it's going to move, well, so then you'll finish translating up here to get that carboxy terminus. You'll get cleavage by the signal peptidase from your signal sequence, and then this part, this stop transfer sequence, will move over into the ER membrane, and that will be your transmembrane domain. And so in this case, your N-terminus is on the luminal side. Yeah?
I think I got confused with the difference between the signal sequence and the transmembrane domain. Is it the same thing?
Sometimes. So signal sequence, when it's most N-terminal, is usually a signal sequence. That's going to be cleaved. It's really about targeting your protein to the ER. But sometimes you might have these internal signal sequences that also act as a transmembrane domain. Those are not going to be cleaved because they're a transmembrane domain.
So when it's external, that's when it gets cleaved.
Yeah, so if it's most N-terminal, then it'll be cleaved. Because if that's your transmembrane domain, you don't have a cytoplasmic, it would just be like stuck in the membrane and hanging. It wouldn't actually have a different part of the protein hanging out on either side if it was that most N-terminal part. Other questions about this? Yes?
What about proteins only on one side of the membrane? The signal sequence ever serve anchored into the membrane and not bring whatever other protein across?
Does a signal sequence ever, so you're saying, does a signal sequence serve as a transmembrane domain, but it's N-terminus? It's N-terminal. I mean, I think anything's always possible. So, I mean, I have no examples in my immediate mind, but I would never say no. Yeah, because that, you never know. Yes?
What did you say the transmembrane domain was again?
Like a definition of a transmembrane?
Yeah.
Okay, so what is a transmembrane domain? It is the part of the protein that goes through the plasma membrane. So that's why I'm talking about hydrophobic, because remember, a membrane is lipid, right? So only hydrophobic amino acids are going to be happy there. And that's why I'm talking about 20 to 30 amino acids, because it's going to form a helix, which needs to be the length of the membrane. Yeah, good question. Any other questions like that? Okay, so then somebody the other day asked if what if you have multiple transmembrane domains so you can get a double pass transmembrane protein, and there you just have more start and stop transfer sequences. Notice in this situation you're dealing with internal signal sequences, as there's no N-terminal signal sequence here at all. And so there's your two transmembrane domains. There's nothing being cleaved because you don't have that N-terminal signal sequence. You could also have even more, the multi-pass transmembrane domains. Here's 7. And so here's your hydropathy plot, right? And so you'll notice you have these peaks, these very hydrophobic peaks, and those are your transmembrane domains. And it would look like this, and it would be like start, stop, and each time you hit one of those helices of hydrophobic amino acids, it would stop and move over into the membrane. Okay? So that's kind of how it would happen. You've got your mRNA and your ribosome and translation and SRP brings it over. That's all still the same, right? Because it's got to get to the ER somehow. And then with each signal sequence, it's going to bind to that translocator, and it's going to pause, and then it's going to move over. And then you'll have more translation, and then it'll bind and pause and move over, and so on until all of them are in there. This all happens in the ER, right? So this is GLUT1, which is a glucose transporter. Okay, so that's enough of that. So there's another thing that happens. There are these GPI anchors. Now it's glycosyl phosphatidylinositol anchored proteins. And so these proteins, this also happens in the ER. And this would be a situation similar to what you were talking about, where you'll have your signal sequence, and you'll basically, not your signal sequence, where you'll have a transmembrane domain that will just anchor the protein into the ER. But what happens is you've got this GPI anchor hanging out here, and then it will get transferred. So you'll actually get cleavage and transfer of the protein to the GPI anchor. So in the end, when you're at the cell surface, you're not transmembrane anymore. It's a protein that's anchored to this GPI anchor. And you might say, why would you do this? What's the purpose? What do you guys think? Why would you do this versus a transmembrane domain? Yeah.
Like a marker?
To be a marker? Well, but why not? So a receptor could be a marker. Right?
You don't necessarily need it to be a receptor to be a marker, but you can just use this as a
So you're saying you could use this in research as a marker? Maybe. What would be the advantage of this versus transmembrane protein? Yes.
Could it be like you don't have that hydrophobic
You don't have to have that hydrophobic domain. Okay. But why would you have this versus? So yes, definitely that's a characteristic of this kind of protein. You're not going to have that transmembrane
Yes,
region.
you have more flexibility on where it could go.
You have flexibility on where it can go. Maybe. There's another hand over here. Well, so yeah.
The protein rating was just going to stay in the ER, but doesn't need to.
If it's going to stay in the air, well, actually, these end up at the plasma membrane. Because all this membrane, all this ER membrane, becomes your plasma membrane. So it's going to bud and travel to the plasma membrane. Yeah. So, well, what if I told you that there are certain enzymes in the plasma membrane that will cleave the GPI anchor? Isn't that interesting? So you can actually release these proteins. Actually, trypanosomes, a parasite, they will release all of their surface antigens this way in response to the immune response. Basically, as soon as they think they're being detected by the immune response, they release all their surface antigens. Smart. Yeah. Questions? Okay, so here's a summary of the delivery of proteins to the ER. Targeting, right, so that would be signal sequence, SRP, association, so that's binding to your translocator, import, obviously import, and then you're going to have folding. So now we're in the ER. So what happens once proteins are in the ER? So these are some main things that happen, right? So you have disulfide bond formation, folding, addition of and processing of carbohydrates. That also happens in the Golgi. Proteolytic cleavage. So that's cleavage of pre-proteins into proteins. Also happens in the Golgi. And then assembly of proteins into different subunits into multimers. Okay, so once the protein's in the ER, it needs to fold, and often that is done with the help of chaperones. One chaperone that is very active in protein folding pretty much everywhere, but there's a family member called BIP. So it's an HSP70, heat shock protein 70. HSP 70, it really just means it's a 70 kilodalton protein that was discovered to be induced by heat shock. It doesn't tell you too much about the function, but most heat shock proteins are involved in protein folding, and that's because when you heat up a cell, proteins tend to misfold, right, or denature. So that's how it was discovered. There are a lot of members of the HSP70 family. One is called BIP. It lives in the ER. Do you think it has a signal sequence, BIP? Raise your hand if you think BIP has a signal sequence. Raise your hand if you think BIP does not have a signal sequence. Raise your hand if you'd wonder what BIP is and why I'm talking about it. I love it. I love honesty. Well, you have to ask yourself, if it's in the ER, how did it get there? Right? How do proteins get to the ER? Signal sequence. So BIP has a signal sequence. And what we're going to learn later is it also has an ER retention signal to keep it in the ER. We'll talk about that later. So BIP is going to bind to the protein as it's coming through the translocator and help it fold. And mostly what it does is it binds the hydrophobic regions to prevent them from aggregating and give it time to fold. Okay? You guys look like you're so excited today. Who comes to my class every morning excited to learn? I was trying to remember earlier. Actually, I believe it was while I was lecturing. This is funny. Do you ever have conversations out loud and then another conversation in your head at the same time? Isn't that weird how that can happen? So I was having this conversation in my head just now where I was thinking about when I took cell biology and whether or not I loved it or if I was excited to learn or any of those things. And actually, I hated cell biology. I hated it because I had a horrible teacher. He wasn't bad. He was just really unengaging. And it was just like this droning sound. So I tried to avoid the droning sound because I know that could be really horrible. But I did horribly in that class because I just never put any of the details together. It never made any sense to me. It's amazing. So all of you guys out there who are feeling that way, you might be a professor one day. Teaching other people who are bored by you. Possibly. Anyways. Okay, disulfide bonds. So another thing, okay, so I'm going through this list of things that happen in the ER. Sorry, I got distracted by myself. Disulfide bonds. Those are basically, they're SS, disulfide bonds. It's an oxidative linkage of sulfhydryl or thiol groups. They stabilize the protein structure. They're involved in protein folding and making that tertiary quaternary structure. That happens in the ER. Why doesn't that happen outside of the ER? Who can guess? Well, the ER is more oxidizing, and so it's it's just a better environment for making disulfide bonds, the reducing environment in the cytosol. Am I right? I always confuse that. Right, the reducing environment in the cytosol often prevents disulfide bond formation. So this is catalyzed by an enzyme called PDI, protein disulfide isomerase. If you're trying to remember the names of these enzymes, you can usually think to yourself, what does it do? Protein disulfide isomerase. Disulfide is right in there, right? So that's an easy one. And so it catalyzes the rearrangement of the disulfide bond. So sometimes you'll have the sulfide the sulfur groups next to each other, and then you'll get this disulfide bond formation, but maybe it'll be in the wrong order for that protein. Maybe it'll just be amino acids that are close to each other, and the PDI will catalyze the rearrangement. And it's doing that by actually binding to and making disulfide bonds with the protein itself. Right. So why do we care about disulfide bonds? It's important for protein structure and function. So an example is an antibody. And does everyone know what an antibody is? Who doesn't know what an antibody is? Who wants me to explain antibodies? Thank you. I love honesty. You make me happy. So when you have an infection and your B cells are alerted to the presence of infection, what do? They secrete antibodies. And those antibodies are going to bind to different antigens or different parts of whatever pathogen is infecting you. Or sadly, pollen, if you're allergic. But so this is the structure of an antibody, and it has two main parts. This part right here, which binds to the antigen. So let's say it's, or vaccine often too. Let's say you have the flu virus. Oh no, let's say you have a infection, a E. coli infection, since that's what we went through last spring. It was really horrible. All of you who had me in 409, it took 6 weeks for my son to get back into school. 6 weeks to clear really bad E. coli. Anyways, don't ever get that. Wash your hands before you eat. The antigens, so there might be different epitopes on your E. coli that are recognized by antibody. Antibody will bind and it will alert the rest of the immune system. Also, you can have neutralizing antibody that will bind to virus and prevent it from entering cells. But pretty much really important in the immune response. And the top part here, it looks like a Y, right? The top part here binds to antigen, and the bottom part down here interacts with other components of the immune system. And so it's a form of communication. And so you'll notice that it's made of essentially 4 protein chains. And they're connected by disulfide bonds. Every time you see SS, that's a disulfide bond. So I put that in because I felt like in the past, maybe people didn't appreciate what a disulfide bond was and why it mattered. But really, it's just about forming the structure of a protein. So then, back to things that happen in the ER, peptidyl-prolyl isomerases. Those are involved with protein folding. They rotate amino acids around the peptide bond to get them in the right, to help with protein folding, to get into the right conformation. Okay, so we've got disulfide bond formation, yes?
Were they resident ER proteins?
These are all, yes, all resident ER proteins, right? Because they're doing their job in the ER. So this is a great example of how proteins need to go where they work, right? So they live in the ER, they work in the ER, you can make the assumption they all have signal sequences. And then you could also make the assumption they all have ER retention signals, which we will get to soon. Yeah, because they need to stay in the ER. So another thing that happens is you get glycosylation. You get N-linked glycosylation. So what you'll have is you'll have your protein being translated in the ER, and as you have an asparagine pop up, you can get this whole block of amino acids, of oligosaccharides that are sugars that are hanging out right here. They will be transferred directly, and that's why it's called N-linked, because the code is N. And that happens on many parts of a protein, and that's really important in quality control. So asparagine, and so are you guys familiar with single letter code for amino acids? No. So when you talk about amino acids, you can have the whole. So I want you guys to all look in your textbook, not right now, at some point, or on the internet, and you'll notice if you pull up amino acid, like just Google like amino acid chart or something like that, and you'll notice that it'll tell you the amino acid name, and then it'll tell you the abbreviation, like ASN for asparagine, and then it'll also have single letter code, and if you go on NCBI to look up a protein sequence, it will usually give you single letter code. And the one for asparagine is N, A for alanine, B for valine, on and on. Q for glutamine. I always remember that because my friend used to say Q-tamine. Whatever gets you through it, right? So this is N-linked oligosaccharide. It's added to most proteins in the ER. It's basically a big block of sugars. And so you can look at this and you'll see that we've got glucose, we've got mannose, and we've got N-acetylglucosamine right here. And so how these sugars look will determine whether or not the protein makes it out of the ER or not. So it's involved in ERAD and protein folding and all that good stuff. Okay, so it's important. And then these sugars are going to be further trimmed in the Golgi. So that's why most cell surface proteins are glycosylated. Okay, here's a practice problem. Therapeutic proteins, such as antibodies, tissue plasminogen activator, which is an anti-clotting factor, and erythropoietin, which is a favorite blood doping drug of choice for cyclists, I believe, right? Who watches the Tour de France? Not just a tour. No cycling lovers here? Not one? Not a one?
I started.
Thank you. Who loves the Ravens? Yeah, of course. I know Lance Armstrong killed it for all of us, didn't he? Okay, anyways. Erythropoietin stimulates production of red blood cells, right? Lots of red blood cells. Okay. So anyways, so all these proteins, I'm totally distracting you, I'm sorry. All these proteins used to be mass-produced in bacteria. So we've got clotting factors, antibodies, and erythropoietin. This process was inefficient and expensive because the protein would never fold right. It would aggregate, and, well, that's not good if you're trying to make money. This problem was solved by changing the host from bacteria to cultured mammalian cells. Why did this change fix the problem? So I want you guys to think about this question, think about what's in common with these proteins, think about what's same and different with E. coli versus mammalian cells, and discuss with your friends. And I'll circulate to expand on that if anyone needs help. Okay, take a few minutes.
just said that we are showing things, but they had it.
A lot of people are calling me over for clarification, and a lot of people have the answer right, which is great. Some people are just wanting clarification on other topics that we've been just talking about, and that's always okay too. But I'm gonna use my list here to see if Austin Gallagher. Oh, there you are. If what your hypothesis is.
I'm saying that bacterial cells don't have an ER or anything like that, so they can't be folded and processed the same way that cells are secreted are, and most of these proteins have to go outside the cells.
Exactly. So these proteins are all secreted proteins. They need to fold properly, but without an ER, you're not getting that disulfide bond formation. You're not having that glycosylation. You're not getting the proper folding. Exactly. Did anyone? Yes.
What about proteins that are post-translation? How do they get that sulfide bonds?
Proteins that are post-translationally imported into the ER.
Yeah, do they just
Well,
do all that once they get into the ER?
well, exactly. It all happens in the ER, right? So, regardless of how you get in there, it's going to happen as long as there is an ER. Right, so that was a question about post-translational versus co-translational import into the ER. Do disulfide bonds and all that processing, is that the same? It's the same as long as you get into the ER. The problem is bacteria don't have an ER. Yes.
So now my question is, so that means that the bacteria are unable to make therapeutic proteins?
Yes, so the bacteria are not very good at making these therapeutic proteins because they're all proteins that are secreted and they need to be processed in a certain way because, well, they do to fold properly.
I guess my question is like for the bacteria's survival,
Oh.
you need antibodies. So
No.
how,
So bacteria do not have a mammalian. So the immune system, as we think about it for ourselves with antibodies and all that, that is very much okay. So now let's go. We're gonna totally go off topic here, but let's think about it. Okay. Do bacteria need an immune system? No. But mammalian cells, specifically, mammalian cells do need an immune system. Why is that? What's the major goal of an immune system?
To get you to reproductive age.
Basically, survival to get to reproductive age, exactly. And it's because you've got a whole organism full of different cells that need to survive so that you can survive. What happens if a bacteria gets infected with something? It dies. Who cares? Because it's one cell. It's in a population. Doesn't matter. I mean, now they do have certain mechanisms for inhibiting viral replication. They do have their own very primitive system for survival, but they don't have antibodies. They don't have this very what's the word? Specific immune response like we have. So yes.
Could it be possible to put microsomes into the bacterial cells in order to process?
Okay, so the question is, can you put microsomes into bacterial cells in order to convert them into good therapeutic protein making machines? You know what? I would never say something impossible because I've been amazed with what biotechnology can do. You just need to be that biotechnologist who figures out how to do that. Because that's a fairly large thing to put into. But hey, maybe if you can express all the genes for making, I feel like you're gonna get stuck somewhere with it. But yeah.
Since there's no ER in bacteria, does that mean that there's no chaperones?
So the question is, since there's no ER in bacteria, does that mean there's no chaperones? No. Actually, the first chaperones were discovered in bacteria. The difference is that there are many types of chaperones, and only some live in the ER. There are also cytoplasmic chaperones and mitochondrial chaperones. Yeah. Good question. Any other? Yep.
If there's no, what is the sec-Y channel then? If there's no ER, where is that in bacteria?
That's in the cell surface plasma membrane.
So they're pushing the proteins out that
In
way?
and out. Yeah.
Okay.
Well, and also you've got, I guess we need to draw out the bacterial cell wall because you've got your peptidoglycan and your inner membrane and your outer membrane and all of that. And so there's that movement.
So that versus the Sec61, it's similar, but the other one's just pushing it into the lumen of the ER? The other translocator?
Both. Sec61, you mean for import of proteins post-translationally? Yeah, so that works differently. Yeah, so that's going to be using HSP70 to kind of pull that protein into the lumen of the ER. Yeah. Any other questions? Okay. I love when a discussion gets started based on these things. Okay. Okay, so let's talk for a minute about quality control. A few people were asking about the glycosylation and what the purpose is, and someone actually pulled me aside and said, okay, you said glycosylation happens in the ER, and that's how a lot of receptors are highly glycosylated. What's the connection there? And I was like, well, no, that's because the receptor starts in the ER and makes its way through the secretory pathway to get to the cell surface. That's that same glycosylation. Yes, it's going to be modified, sugars will be trimmed, but you will have glycosylation that originates in the ER and is modified in the Golgi that is present on your membrane protein on the cell surface. And so then that person asked me, well, what's the purpose of putting these sugars on? And I said, well, we're going to talk about their role in quality control. And so we'll talk about that for a minute. This is specifically talking about two different processes, ERAD, which is ER-associated degradation, and the UPR, unfolded protein response. People always get confused between the two, think they're one, swap back and forth different components. So I just want to tell you in the beginning, they're two different things. Try to remember that they happen under different circumstances. Right, so only properly folded and assembled proteins can leave the ER. You don't want misfolded proteins leaving the ER, right? That's bad. Right, if a misfolded protein gets to the cell surface, well, then you might have a receptor that can't do what it needs to do. That might affect the whole signaling pathway, right? So you're going to be retaining a protein to be retained in the ER until it's properly folded, and if it's not able to be folded properly due to a mutation or something like that, then it will be removed through ERAD and degraded in the cytosol. Okay, so proteins that cannot be folded, they're removed by ERAD, ER-associated degradation. And the unfolded protein response is induced by an excess of misfolded proteins in the ER. So some drugs, for example, cause a lot of ER stress. They cause an accumulation of unfolded proteins, and that will activate the unfolded protein response. So that's different from ERAD, which is just scooting the bad ones out. Like, oh, this one's not folding. Retrotranslocated it through that translocon. It gets ubiquitinated, recognized by proteasome, and degraded. Different story. So in the unfolded protein response, this activates this response, which increases transcription of proteins that assist in proper folding. What kinds of proteins assist in proper folding? Chaperones. And there's other ones too, but that would be your most obvious answer, yeah. So chaperone, peptidyl proteolysomerase, PDIs, all of these proteins we just talked about. It will increase transcription of these proteins so that they can be made. They can go to the ER and help to deal with this large amount of misfolded or unfolded proteins. Okay? So you might say, okay, you were talking about glycosylation and how it's involved in protein folding, and tell me how this happens. And so there's two players that are important here, calnexin and calreticulin. So they are ER-resident proteins that are involved in retaining misfolded proteins in the ER. And what they do is they bind to these sugars. So here's your protein. Remember, it was being translated through your translocator, and then it was being N-glycosylated, glycosylated with these carbohydrates that look like little claws. And then you've got calnexin. Calnexin is a membrane protein. It's a membrane protein that lives in the ER. It never goes, well, if all goes well, it shouldn't go to the plasma membrane. It lives in the ER. It functions in the ER. And its job is to hold onto those carbohydrates to retain it in the ER while it's folding, so it doesn't go out. And then calreticulin is also involved with retaining and aiding in folding, keeping the protein in the ER until it's properly folded and assembled. So this figure here is showing the folding of hemagglutinin, which is a flu gene, and it's a trimer. And so it needs to fold, and then it needs to assemble into a trimer. Remember, in the ER, multi-subunit proteins will assemble, so this is an example of multi-subunit proteins assembling. And calnexin and calreticulin will retain these proteins in the ER until that's done. And then it can go and do what it needs to do. Okay? Questions? Okay, so go a little bit more detail. So you might say to me, okay, so calnexin and calreticulin are binding to these sugars. How does the protein ever get out of the ER? And so there are these enzymes in the ER that participate in this folding cycle, which basically what it's doing is it's cleaving sugars and adding sugars, cleaving sugars and adding sugars, and that's regulating the binding of calnexin to the protein during the folding process, right? So you'll have an unfolded protein, and it'll have that sugar, that block of sugars added. And so you notice that what's going to happen here is you've got these 3 glucoses here, and then an enzyme will come along and it will trim the 2 of the glucoses off, and so that will allow it to bind to calnexin. And then they'll be folding, and then you'll have another trimming, which will remove that last sugar, so then it can't bind to calnexin. And so if it's properly folded, it can leave. And it can bud and go on to the Golgi. If it's not properly folded, then another enzyme will come and it will add sugar again. And it'll be bound again, and it'll participate in folding again. And it'll get cleaved off, and it'll so on until either the protein's folded properly and it can leave and bud out of the cell in a vesicle to go to the Golgi, or it will be modified in a different way with different sugars. I believe cleaving of a mannose, but it doesn't matter. And then it is targeted for ERAD. And then it's retro-translocated, so meaning it's translocated backwards out of that translocon and recognized, ubiquitinated, and degraded by the proteasome. So you could think of it as a folding cycle that's regulated by the glycan code. Glycan meaning sugars, and it's kind of a code, right? The shape of these sugars determines whether calnexin can bind or not. So that trimming and adding sugars is regulating whether or not it's going to be retained in the ER or it can go. Questions about this? Okay. We used to do a paper on ERAD. I think I got rid of it because it overwhelmed people, but it's so fun. Okay, so a protein that's irreversibly misfolded, so you can go on forever and ever with this cycle of folding. It will never fold properly, possibly due to a mutation, such as the cystic fibrosis. Cystic fibrosis, what's the protein? Come on, guys. Tell me, what's a protein that's mutated in cystic fibrosis? It's a channel, calcium channel. Anyways, there's like a mutation of one amino acid that basically activates ERAD. And so actually that's perfectly functioning, it's actually fairly well functioning, the protein, but it's just activates ERAD and is degraded. And so you have cystic fibrosis. That's one major mutation. There are a number of mutations, but that's a very well characterized one. Okay, so if you're irreversibly misfolded, you'll have trimming of the sugars a different way, and then you'll get retrotranslocated out, and then you will get ubiquitinated and degraded by the proteasome. How are we doing here? Okay, 5 minutes, guys. Stay with me. Yes. Oh, I know. Tell you all about it. I just got, thank you. I planted him right there. What's ubiquitinated? Well, ubiquitin is the best protein ever. Just write that on the exam. Best protein ever. No, so ubiquitin, it's a small protein, and it's covalently added to other proteins. So it's a post-translational modification. Post-translation, so it's added to a protein. and it is conjugated to a lysine. So specifically, it's added to a lysine on a protein, and it does many things. But basically what it does is it changes the shape of the protein in a way that makes something happen. And the most obvious one that we've all learned about a million times is recognition by the proteasome and degradation. So if you want a protein to be degraded, if the cell wants to degrade a protein, it's going to ubiquitinate it. It's going to add a ubiquitin molecule to it, and then another and another and another, because it's a polyubiquitin chain, and that polyubiquitin chain will be recognized by the proteasome and degrade. This is really important because you don't want, the proteasome can't just be willy-nilly degrading everything, right? You only want specific proteins to be degraded. So there are different kinds of ubiquitin chains that mean different things. So the one that we are talking about in this case that sends proteins to the proteasome to be degraded is a lysine 48 linked polyubiquitin chain, but there's also lysine 63 linked polyubiquitin chains that are involved in activating signaling pathways. You can have monoubiquitination, which is involved in endocytosis, involved in gene silencing. Also, you can have lysine 63 linked polyubiquitinations involved in the DNA damage response. Lots of good stuff. So I'll let you go here, but we'll start with ubiquitin on Tuesday.